Morning. On behalf of the International Society of Heart and Lung Transplantation's Research and Immunology professional community, I'd like to welcome you to our webinar entitled Sequencing the Future. Since the year 2000, the cost of DNA sequencing technology has decreased by more than 300,000-fold. We are only beginning to understand what can be done with next-generation sequencing technology. Today, we'll hear from an international team of thought leaders about the promise of sequencing-based technologies to improve thoracic transplantation. Today's session will include 4 10-minute talks followed by 15 minutes of panel discussion. I'm John Greenland, a pulmonologist, associate professor of medicine and lung transplant researcher at the University of California, San Francisco. And I'd like to introduce my co-host, Adriana Zevi, who is a distinguished pathology service professor of surgery and immunology, and the director of University of Pittsburgh's Medical Center Histocompatibility Laboratory.
Thank you, John. It is my pleasure to introduce the first speaker, Dr. Brendan Keating, who is an associate professor of surgery at Perelman School of Medicine, University of Pennsylvania. He is well known for his expertise in large-scale genomic and genome-wide association within the transplant setting. He will speak to us today about the genomics, leveraging the genome to guide donor matching and immunosuppression. Brendan, it's yours.
Good morning, everyone. So I'm going to talk today about some efforts going on over the last few years to help guide donor matching using genomics and also for immune suppression reduction therapy as well. So it's been about 20 years since the human genome was sequenced at a cost of somewhere between $2 to $3 billion. And it really did set up a whole industry downstream that's been looking at the transcriptome all the way to infectious disease to other large-scale sequencing efforts. And over the last few years, there's been a big endeavor to really try and link up electronic medical records with patients' DNA samples and biological specimens. And this has really helped propel medicine along just in the last few years. So this was an article by Eric Green about 12 years ago. I basically tried to map out how the human genome would impact society downstream. And you can see at the bottom here, we're really starting to get into the point now of improving patient outcomes and better healthcare. So if we look at the old classic HLA typing, all the way down to 4-field resolution, this has really been superseded in the last few years with the use of applet matching, and also a whole range of different donor-recipient interaction methodologies have been coming into play. If we look beyond the HLA class 1, class 2 region, the rest of the genome is very important for transplant outcomes, but we've just never really had the tools or the numbers of individuals to really start dissecting these interactions. So this was something we published a couple of years ago in the Lancet, where we looked at about 500 kidney transplant donor recipient pairs, and they were basically matched for applet mismatch in this analysis. And we looked at roughly 60,000 variants across kidney transmembrane proteins. And we looked at how these individuals, after adjusting for HLA applet mismatch, how the graft survival looked at 5 years and 10 years. So basically, the more mismatches you had, the worse your survival was at 5 years. So basically, if you were in the least mismatched quartile, you had about 98% survival, all the way down to 82% if you're in the worst mismatched quartile. In this study, we also went on to actually show alloantibody against some of these protein coding changing variants. So we did this using a peptide array where we physically printed peptides, roughly 170,000 peptides. And this shows you here some some work we did looking at heart transplant alloantibodies. We can also look at polygenic risk scores. So these are basically, we take the known variants that impact something like type 2 diabetes. We can look across the genome and use weighted risk scores to put someone in a risk category, and we can combine that with clinical covariates as well. So this was something we published last year in Nature Medicine, where we looked at the polygenic risk score for type 2 diabetes in liver donors and recipients, and also in kidney donors and recipients. We're basically able to show that you transfer the genetic risk for post-transplant diabetes in the liver donor, but not in the kidney donor. The recipients do have independent risk for post-transplant diabetes. So this could actually impact donor selection, because especially in the pediatric living donation setting, if all things were equal between donors and you had, say, 3 living donors that you could select from, in that case, those with the least mismatch score, they could give a much longer lifetime for the allograft. So this is just one example. And then if we look across some of the other polygenic risk scores, say for dyslipidemia or for chronic kidney disease, are you transferring risk either in the living or deceased donor setting? So I think in the next few years, you're really going to start to see these type of things impact donor selection, especially donor cause of death as well. There's a lot of diseases such as stroke that impact that. So as part of a UO1 study, we've been looking at donor-recipient interaction using HLA and non-HLA in a variety of different kidney, liver, and also lung and heart transplant studies across the globe. So we also have been doing a fair amount of work in the immunosuppression reduction arena as well, particularly in the liver transplant setting. So again, leveraging the power of the genome, we looked in 2 studies, the CTOT3 and also the ISHLTv study, to look for biomarkers impacting rejection, so basically to diagnose and prognosticate rejection. In the ISHLTv study, this is a very special study where people were reduced in immunosuppression after 12 months with a biopsy to ensure there was no subclinical rejection. And basically about 70% of individuals could tolerate 50% reduction of immunosuppression. So we basically started looking at microRNAs, and we could show that individuals who had a biopsy-proven rejection, they had elevation in a number of 2 microRNAs, but after regression, and these basically predictive rejection as well as diagnosed rejection. And we think that this will basically move on to being able to help reduce immune suppression using this guided method. So if someone is on 50% immune suppression, we'll just continue to monitor them to ensure that they stay in the blue safe zone. There's also been a lot of work done in the tacrolimus genotype guided dosing, particularly in African Americans. This is work out of DCAF Genomics, which showed that based on CYP3A5 genotyping, that you could help a starting dose of tacrolimus. All of this still has to be proven at scale. There's also been a lot of work in trying to reduce immune suppression in the kidney setting using epileptic mismatch as well. And then just to finish up, a lot of work we've been doing is combining genomics with transcriptomics, proteomics, metabolomics, and basically trying to look in prospective samples across individuals to try and delineate how the donor impacts these outcomes in these subjects. And we've also been doing this in the human and pig xenotransplant setting as well, and again, leveraging the power of genomics. And I'd just like to thank my collaborators in the IGene train and the sponsorship from NIH for these studies. Thank you.
Thank you so much, Brendan. Now I'd like to introduce Phil Halloran, who is a professor at the University of Alberta, director of the Alberta Transplant Applied Genomics Center, the CEO of Transcriptome Sciences and the former editor-in-chief of the American Journal of Transplantation. As a world leader in transcriptome-based diagnosis, he's going to be speaking about how we're going to bring these molecular diagnostics to the clinic. Thanks.
Can you see my screen?
Yes, we
I'm
see.
sorry.
Yes, you see the thank you slide.
Okay. Okay, you should be seeing a screen starting with molecular microscope.
Yes, looks great.
So our interest has been in reading the tissue. Our main objective is to take relatively small amounts of tissue, because this is all in clinical trials .gov regulated environment, and so we're very strictly limiting the amount of tissue. So the diagnostic system will eventually have to find out exactly how much tissue we need to avoid sampling error, but I'll show you what we have. This is the team that works on it, and we are grateful to a variety of sources of support, including the Mendez Foundation, Genome Canada. So we study endomyocardial biopsies. We're looking at the at rejection, injury, and injury-rejection relationships. We're then trying to do the same thing with lung transplant transbronchial biopsies and mucosal biopsies. And mucosal biopsies, we're trying to tease out the relationships with clad, and of course, identifying the ABMR signal in the TBB and in the 3BMB. Long story short, we identify an ABMR-like NK signal in TBBs now, and that'll be presented at the ISHLT meeting in April, and it's under review. And we see we have identified clad-related molecular signals. And then I won't have time to talk about it today, but we're engaged in the trifecta studies, which are studies to relate cell-free DNA to the molecular phenotype of the biopsy. There's a number of publications here. Well, I won't go into more detail at that point. So the general approaches we have to understanding the tissue is to discover the underlying injury states and recalibrate the existing diagnostic systems, make MMDX available, and so it's licensed to Thermo Fisher. But this is, I'm really talking about a university research project. The licensing is not really relevant here. And then we want to compare MMDX biopsy findings to other biomarkers. And that's the basis of the trifecta study, in which we're looking at MMDX versus DSA and other antibody measurements, and also cell-free DNA. We can actually extend this to other types of biomarkers as well. So the trifecta studies have been published for kidney, and the first generation reports for heart will be presented, we hope, at the American Transplant Congress in June. Now, our interest in tissues is that biopsy if necessary, but avoid if possible, but use the biopsy maximally. It's the major site for discovery of disease mechanisms. It's also the site for definitive diagnosis. And then we want to be able to infer diagnosis without doing a biopsy if necessary. So if you have to do a biopsy, optimize the utility, and we would suggest that get the molecular phenotype. We use microarrays. The microarray we use as it's a very standard platform with high quality and high reproducibility. And so we can accumulate reference sets over many years, and you know that we're able to normalize the result of any new biopsy and add it to the reference set. This is a very critical point. So that's looking at the rejection and the injury states, using messenger RNAs from 50,000 probe sets to infer the signal for 19,000 genes. So the process is that you stabilize the biopsy in RNA. Later, you send it by FedEx. The biopsy is then extracted to generate the patient's RNA, which is then labeled and hybridized to the chip, and this generates a vast spreadsheet, which we turn with our software into an MMDX report. So for an endomyocardial biopsies, we're looking at the rejection and injury states, and I'll just go through this briefly. These are the centers that have been collaborating with us. We're very grateful to them, especially during the pandemic. So we're able now to identify T-cell mediated rejection and antibody mediated rejection. And also recent injury, which is in heart. Heart is very pro-inflammatory when it gets injured. And so you get a macrophage infiltrate and we can pick that up. So this is the report. I think many of you are familiar with it, but it's published. So in this biopsy, we have, it's not the best biopsy we've seen, but it has no clear rejection. And so it's right in the center. Panel 1 is principal component 2 versus 1. And the distribution of the biopsies in this principal component analysis is antibody-mediated rejection, T-cell mediated rejection, and no rejection. And then in principal component 3, injury separates out in principal component 3 due to the macrophage infiltrate. All right, so right now what we're looking at is this is 1,320 biopsies. We actually are working now with 3,200 biopsies, but this is the 1,300 samples. So we get no rejection, and then varying degrees of rejection is coming in here, and then antibody-mediated rejection and T-cell mediated rejection, and then this is the detail of the principal component 3 picture with the recent injury here. Now, what we end up with is 2 vectors then in terms of rejectionness, the ABMR vector and the TCMR vector. And the real question continues to be, what's going on in these biopsies that have some abnormalities, but are not clearly related to the disease phenotypes? We've spent a lot of time on this, and it does seem that there is something that lies beneath. So there's a mild antibody-mediated rejection-like state that influences outcomes that we're still not diagnosing, a lot of it DSA negative. When we look at the DSA state, we find that only 66% of the ABMR is DSA positive, and that's about the same now in kidney as well. The DSA negative is more in lung. In the transbronchial biopsies, most of the ABMR signals we get are in DSA negative cases. And that's probably because the vast microcirculation of the lung, and for that matter, the liver, we think it absorbs the antibody and removes the signal from the circulation. So that means that you really do need to recognize DSA negative antibody-mediated rejection. The odd thing is that if you look at the failures in the population, the most intense DSA positive, most intense antibody-mediated rejection is whether it's DSA positive or negative, doesn't actually cause graft failure, at least rapidly. It causes slow fibrosis. TCMR, though, is rapidly associated with graft failure. It's a devastating disease for the parenchyma. So, then we're also looking at the injury states, and what we see is the emergence of a late injury atrophy fibrosis state. In about 20% of the heart biopsies that we receive, we see a late injury state. It impairs prognosis. It's only got rejection active about half the time, but it's probably being called CAV a lot of the time. And that is present, it's probably a problem in a lot of the long-term hearts, and it's probably under-recognized right now. And it seems to be an atrophy fibrosis state, similar to the atrophy fibrosis state we see in other organs. So when we distribute the injuriness, we get no injury, mild injury, moderate injury, and most of this is the time course of the early post-transplant injury. And then severe injury. Severe injury and late injury are where we see our failures. So this is these are hearts that are at more risk. We're also working with a survival classifier in hearts. So these are the 2 main dimensions of injury that we understand in hearts: the recent injury and the severe deterioration, the late injury with deterioration. Okay, left ventricular ejection fraction correlates more with the injury than with the rejection. So this is the uh, left ventricular ejection fraction abnormality on this scale. And it's the late injury and the severe injury which have the abnormalities. And is it related to CAV? Probably. But in fact, in our studies, we have not had detailed coronary phenotyping. And then how is rejection related to injury? Well, it seems that T-cell mediated rejection produces severe injury and late injury and has almost never got normal parenchyma. Antibody mediated rejection has a spectrum. It doesn't usually produce severe injury, but the late antibody mediated rejection is associated with the atrophy fibrosis phenotype. So it's a slow deterioration, which is well tolerated, probably for years. And in terms of survival, the histologic rejection does not predict survival. MMGX does, but it's the TCMR and the injury score that predict. And in terms of injury phenotypes, the strong prediction is in the severe and late groups. Now in lung transplantation, I'll just briefly review this. We're looking at the transbronchial biopsies and the mucosal biopsies. And the mucosal biopsies, Kieran Halloran and Shafka Shavji worked out a way of biopsying the third bronchial bifurcation in a standardized way. When you're doing biopsy studies, the biopsy has to be standardized in order to normalize it across a large population. And these are the centers working with us. We're again very grateful to them. So this is the interlung study. And again, it's all clinicaltrials.gov. And these are the initial reports. So we're seeing basically in the transbronchial biopsies, we can mainly read T-cell mediated rejection. What we've been recently doing is try to understand this second dimension and probably the antibody mediated rejection. And again, this is under review, so we will be presenting it. The antibody mediated rejection presents as a set of NK-rich biopsies in this region. And it seems to be well tolerated as it is in heart and in kidney for a period of time. The clinical significance of antibody-mediated rejection is based on the NK phenotype. It has to be determined. So it's NK and it's also gamma interferon. Those are the features of antibody-mediated rejection across all the organs. The T-cell mediated rejection is typical, and in lungs, it's devastating for the short-term outcomes. I'll just move. This is the mucosal biopsy report that we're working on. It has many of the same features, but it does not represent the microcirculation well. So we'll probably have to define antibody-mediated rejection in tissue which is rich in microcirculation, which in lungs is the alveoli. So we can detect antibody-mediated rejection. So now CLAD has emerged as an interesting signal. CLAD has emerged as a signal which has got some features of active deterioration, which is also seen in kidney transplants. So we're able to move the signal into the kidney transplants, and this is the signal we're getting. This is in the transbronchial biopsies. It's an active injury signal. It's got some very interesting features like IGF and HIF1 alpha. But this signal predicts outcomes. Interestingly, CLAD as an outcome, it's got a devastating effect. It's like advanced metastatic carcinoma. And the molecular phenotype isn't as predictive as the physiologic phenotype, but it is independently predictive. And there's also a lot of dedifferentiation of the transbronchial biopsy. These are the transcripts of the normal lung, which are lost during CLAD. And the main feature of it is injury. So these are our injury gene sets that are increased in CLAD, and dedifferentiation. These are the normal lung transcripts which are decreased in CLAD. I'll just rapidly look at the CLAD-associated genes in transbronchial biopsies. We just suggest that, that this is an advanced injury state in which the tissue is probably committed to deterioration. And that's what we also see in kidneys. So in the In the airway mucosa, we've got a similar signal, some differences as you'd expect, but the principles are the same. It's an injury signal. Some sharing of the molecules between the mucosa and the transbronchial biopsy. We got genes like LOXL1, which is a lysyl oxidase enzyme, which is seen in deteriorating kidneys and deteriorating transbronchial biopsies and deteriorating mucosal biopsies. Let me just advance quickly and show you. This is the LOXL1 signal in the transbronchial biopsy as well. I just showed you the mucosal biopsy, so it's elevated in clad. And this is the picture in kidney transplants. And some of the genes, actually, this is the genes from kidney transplants. And some of the genes that are the highest associated genes in kidney transplants are also elevated. In deteriorating kidney transplants are also elevated in 3BMBs and TBBs. This is an interesting analysis. This is the survival of kidneys predicted by the clad genes. And this is in particular, the top clad genes predict deterioration of kidney transplants in order. And LOXL1 predicts the deterioration of kidney transplants, just as it is associated with clad in TBBs and 3BMBs. So that's really where we are. Our goal is to understand the molecular phenotype of disease, to license it as a diagnostic system where that's applicable, to guide clinical trials, to use this for targets for intervention, and to understand the relationship between the molecular phenotype of the disease and the non-invasive biomarkers. So thanks very much for your attention.
Thank you, Dr. Halloran. It is my pleasure now to introduce Dr. Teresa Martineau, Associate Professor at Toronto General Research Institute, also Toronto Lung Transplant Program, University Health Network. Dr. Martineau is known for her contribution to understand the pathogenesis and mechanism of chronic lung allograft dysfunction. She will speak to us today about the single-cell technologies, the potential for the single cell and the spatial transcriptomics to guide rejection diagnosis. Teresa?
Thank you so much, Adriana. Can you hear me? Hopefully it's working. So hi, everyone. It's a pleasure to be here today to talk about single-cell transcriptomics. In terms of disclosures, I have received Sanofi support for some of the single-cell transcript analysis experiments that we're conducting. Let me start with the fruit and smoothie analogy, if that's okay, to explain single-cell analysis. So in bulk RNA sequencing, which has been published for many years already, you take a bunch of cells, you grind them together and analyze all the transcripts mixed together. That's the smoothie. You can tell that there was probably some watermelon, lemon, strawberries, but you're not sure about the kiwi or the dragon fruit. Single-cell transcriptomics is more like single fruit analysis. So you can tell now that your pile of fruit included half a watermelon, 2 lemons, no dragon fruit at all, etc. I like this depiction from Dan Chambers' review from 2019. You can see how bulk RNA sequencing gives you an average gene expression profile, but you can't differentiate the individual cells. In single-cell sequencing, you get the actual transcripts for each individual cell. High-throughput single-cell RNA sequencing didn't become possible until around 2015, when in-droplet cell packaging and microfluidics became possible. And this is a simplified version explaining the workflow overall. So one needs to start with a single-cell suspension here. This is a relatively easily obtainable for body fluids and blood, but a little bit more complicated from organs and tissues. The cells are flown through the microfluidic system and combined with barcoded beads, and then they're trapped in these oil droplets. The transcriptome of each cell is then generated inside each droplet and specifically barcoded to that cell. The transcripts can then be combined and sequenced, and the sequences can then be assigned to the cells based on the barcodes. This generates pretty large data sets that require pretty powerful bioinformatics. I want to mention that the cleaning of the data is really important to remove dead cells, doublets, ambient RNA, etc. And then you generate this final Disney or UMAP plot that's often seen in publications that shows cells, individual cells that are clustered based on their similarity, based on the thousands of genes inside each cell. So as you likely know, the number of publications related to single-cell RNA sequencing has been increasing over the years. Here's an approximate number of those related to heart transplantation. These are all mouse model derived, as far as I could tell based on my search. And then these are the lung transplant related ones. In the next few slides, I'll just show some examples from human lung transplant single-cell RNA sequencing analyses. So Mark Snyder was one of the first authors publishing on single-cell RNA sequencing in lung transplantation, focusing on tissue-resident memory T cells. This is his actual follow-up paper with John McDyer, where they looked at T cells and T cell clones in the bronchoalveolar lavage, or BAL, of lung transplant recipients with rejection before and after treatment. So really neat cohort. This is a good time to mention that most single-cell RNA sequencing uses the 3' assay, meaning that the mRNA is reverse transcribed starting from the 3' end. However, the VDJ sequence of the T cell and B cell receptor mRNA is actually near the 5' end, so 5' sequencing can then combine gene expression profiling with the concurrent sequencing of the B or T cell receptor, which is really powerful. So in this UMAP on the left, you can see 10 T cell clusters, so quite large heterogeneity of T cells. And this other UMAP shows the most common T cell clones or TCR sequences. And you can see that the most common clonotypes showed in color here map to cluster 0 and 1 here on the left-sided UMAP, so these T cell clusters. And these, based on these feature plots, you can see that they express CD8, granzymes, perforin, interferon gamma. So all this data together suggests that these clusters represent oligoclonal expanded cytotoxic CD8 T cells, which expand during rejection and, as the authors further showed in the paper, actually remain present after treatment. So pretty powerful data to glean from this analysis. Switching gears a little bit, here's an example of a more exploratory type of analysis performed by Sajjad Moshkal-Gosha and led by Stephen Jouvet in our Toronto group. They performed single-cell RNA sequencing on BAL cells of 6 lung transplant recipients. And this t-SNE plot shows macrophages only and shows an impressive heterogeneity of the macrophage populations. The patients with acute lung allograft dysfunction actually have 2 macrophage populations that were not present in the stable patients, and these were the interferon-stimulating gene-expressing macrophages and the metallothionein gene-expressing macrophages. This is really just transcript data again. So in subsequent analyses, Sajjad and Stephen validated their findings using flow cytometry and in vitro assays to show that these macrophages truly have unique pro-inflammatory functions, not just transcriptomes. Another very useful feature of single-cell RNA sequencing is the potential to deconvolute donor from recipient cells. Byrne and colleagues used sex mismatched transplants and used male and female genes to distinguish the two. In more recent analyses, Gavin Wilson and John Young at our center have worked on using single nucleotide variation differences between donor and recipient to deconvolute these. Both approaches actually show that the donor alveolar macrophages get replaced by recipient alveolar macrophages over the course of about 2 years. So up to now, I focused on the bronchoalveolar lavage from our patients, which is a liquid and already comes with a single cell suspension. Our group was interested in assessing cells from the actual lung allograft tissue as well. Alan Duong, a PhD student in my group, has been working on optimizing the dissociation of the tissue and isolating cells from lungs explanted at the time of retransplantation for chronic rejection. This UMAP here shows the, again, large heterogeneity of cells within the lung tissue. And the subsequent t-SNE plot here shows macrophages only, and we were able to identify within chronically rejected lungs the same macrophage populations as were present in the BAL. So just a little bit about spatial transcriptomics. I showed you how we could go from smoothie to single fruit analysis. Now, what if you want to know where exactly the fruit is located in the orchard? I haven't found any examples of spatial transcriptomics in thoracic transplantation, so we'll stick with kidney for this example. So in this analysis, the authors wanted to address the fact that acute rejection is a focal process. They used this whole genome profiling platform to study 5 tubular and 3 glomerular regions of interest in kidney biopsies that were derived from patients undergoing T-cell mediated rejection. And the authors found that the tubular areas had upregulation of genes that were associated with T-cell activation, differentiation, and proliferation, but also kidney development. And they really concluded that these genes were not previously associated with kidney rejection and also were not associated with the Banff scoring in the same regions. So they found that the data was quite novel and they would not have been able to get at it if they didn't have this spatial component to the analysis. So the question that was included in my title was whether single cell and spatial transcriptomics can guide rejection in thoracic transplantation, guide rejection diagnosis in thoracic transplantation. And I hope I can convince you that it's a yes. You know, we really want to know more. We always want more data. We want to know what the individual fruits are, where they're located. In summary, single-cell RNA sequencing really can give us very detailed info about individual cells, their heterogeneity. You can really get it from many sample types that are available from the post-transplant timeline, and you can glean information, at least indirect information about cell function. And sometimes you can even get this at the spatial resolution. There's some important limitations. You know, the sample prep can influence your results. It's still quite costly. And you don't have protein and function data. You really need validation to confirm your findings from single-cell RNA sequencing. I'd like to acknowledge all the people at my center who've been working on related projects and supporting this, and also everyone around the world who's contributing to this advancement of knowledge. Thank you so much.
Thank you, Dr. Martin. Now it's my pleasure to introduce Sean Agbar-Eno, who is a pulmonologist at Johns Hopkins University School of Medicine and the Lasker Clinical Tenure Track Investigator and intramurally funded Distinguished Scholar at the National Institutes of Health. He directs the ISHLTv Applied Genomics Research Group and has pioneered cell-free DNA for lung transplant diagnosis. He will tell us about what's coming next in cell-free DNA analyses. Thanks.
Hi, just a minute. I am having some technical difficulties. Need to turn my video on. Easy. Bear with me. Video on. Okay, thank you, John. Let me start with my disclosures. I would start with number 3. I don't have financial compensation from companies that collaborate or lead studies that are run by companies, some of which are cell-free DNA related companies. And I want to start with this slide, which is truly appreciating the people who have been so fortunate to work with, that run a lot of these programs and run a lot of this assay and build this infrastructure, and that give me the privilege to be able to stand here and talk to you. So that's the NIH lab here, not the consortium, but the lab at the NIH. Cell-free DNA, I think it's becoming familiar now in our community. These are short fragments of DNA that are released into the circulation. And the studies that I'll show here, you're using plasma. And if I can just give you one take-home message here, the idea here is that with a vial of plasma, can you tell what is happening at distant organs, like allografts that have been transplanted? And can you be a little bit more precise, just looking at plasma and plasma DNA? So that's cell-free DNA, quite abundant in plasma. The first generation test that I would show here, I will not talk a lot about it, is based on single nucleotide polymorphisms, as our group and many other groups have shown, that if you use that first generation test, targeting SNPs to track allograft-derived cell-free DNA, you see that the cell-free DNA is elevated at the time of rejection. However, several weeks to months before that episode of rejection, particularly in the case of AMR, you have elevations indicating that cell-free DNA could be used for early detection. So the assay has really good sensitivity, but its specificity in differentiating types of rejection or differentiating rejection from infection is very low. So good sensitivity, low specificity. So next step, could we find a more specific test? And if you think about it, but maybe that answer is yes. Maybe, I use maybe really carefully here. Let's look at the rejection, acute cellular antibody-mediated rejection. As we know, the cell types and the tissue types that are involved in these two types of rejection are different. It's true that in the lung, there's quite a bit of overlap between the two types of rejection, but here I'm using slides from kidney and heart. In the acute cellular rejection, lymphocytic infiltration within the parenchyma of the lung versus neutrophilic infiltration for acute cellular rejection. So different cell types, different tissue types contributing to injury. The question then becomes, if we know that, and we know that each tissue have tissue-specific epigenetic fingerprints. Take DNA methylation, for example. DNA methylation of the brain versus the heart versus the kidney, they are distinct. That's what guide, it is these epigenetic fingerprints that gives the organ its identity and its function. So if we know that, and a library of most of these have been cataloged. So if I know that, can I then do cell-free DNA epigenetic analysis and scan cell-free DNA for this epigenome? Could I provide a whole body injury map and be able to define disease a little bit better? Well, the two approaches that we've tried. The first is cell-free DNA methylation analysis, or CF-methylome for short. And I'll show you a case where these have been applied in COVID-19. So this is work that was published early on in the pandemic. If you look here, these are two patients with COVID-19 who died. Cell-free DNA is high at the time of admission. It decreased over time. Between day 10 and day 15, these patients go into an inflammatory phase, exponential rise in cell-free DNA until they eventually die. You can see the catalog to your left there shows you the different tissue types that are producing the cell-free DNA. Again, these tissue types identified using tissue-specific signatures of DNA methylation that are preserved on cell-free DNA. Compare that to patients who died, you see different patterns. Indeed, the use of cell-free DNA could identify, at the time of testing, could identify patients who need ICU care with an accuracy of over 97% in COVID-19. We've shown the same in pulmonary hypertension. This is work that was led by Sam Busker, who is now at UCSF with hanging out with John Greenland. You could see here just different tissues. If you look at the tissue map on the left-hand side, healthy controls, which are all here in purple, they're all clustered to your right. And these are unsupervised clustering. And then more to your right, if you look on the top here, is more yellow and red, which are patients with moderate risk or high risk pulmonary arterial hypertension. Again, different tissue types predicting different risk. If you look in these patients, whether it's COVID-19 or pulmonary arterial hypertension, you can then identify the tissue sources of cell-free DNA that predict survival. In pulmonary hypertension, for example, you take patients with low levels, you have a survival like this. Patients who have moderate levels of that cell-free DNA, you have lower survival. And if you have patients with high levels of cell-free DNA, the survival goes down. So in these diseases, using plasma, you can then identify tissue injury patterns that are biologically plausible and that correlates with disease severity and transplant outcomes. So that's step one. Now, what if you want to try something else? As we know, most diseases have endotypes. I think Phil just presented different endotypes or different types of injury patterns that you can see. So there might be need for molecular information about your disease structure. And can you get that using plasma? Take lung transplant patients, for example. About 50% of them will make DSA. And out of that 50%, half of them, so 25% of the total, approximately 15 to 25 will develop AMR. So these patients with DSA, then you can see they're characterized by some who will have AMR, some who will not. Even if you look within the AMR cohort, there's some who will respond to treatment and some who will not. Can you identify these kinds of differences in your disease structure using cell-free DNA? Maybe you can. Now, another approach, cell-free DNA chromatin immunoprecipitation. This concept was introduced by a group from Israel, published in Nature Biotechnology a few years ago. We've adapted that in the lab and asked a very simple question. If chromatin immunoprecipitation is supposed to be a proxy for gene expression because chromatin modifications drive gene expression. If I therefore use cell-free DNA, chromatin immunoprecipitation, am I able to pick up genes that I know are expressed? The answer to that is yes. Let's look at housekeeping genes. As you're familiar, housekeeping genes are genes that are expressed all the time. They're constitutionally expressed. There are about 9,100 of those genes that you could see here in red. If you do cell-free DNA chromatin immunoprecipitation or CF-ChIP-seq, you can precipitate about 91% of those genes consistently in every subject that we looked at. And these were 5 different patients. We've shown the same in heart transplantation. So you can precipitate genes that are expressed, and we've looked at that in different disease states. Next question, can I identify differential gene expression or gene signals between 2 states? Here we looked at healthy controls and heart transplant patients who are taking tacrolimus, a calcineurin inhibitor. And if you look here, between heart transplant and healthy controls, a majority of those genes are downregulated, as you would expect in these patients who are taking immunosuppression drugs. Asking the question, these patients are on tacrolimus, what is happening to calcineurin-related genes? If you look here, they're downregulated in these patients and differential between heart transplant and healthy controls. So I'll stop here. Some take-home points is that the SNP-based approach for cell-free DNA methylation, SNP-based approach is sensitive to pick up rejection that is non-specific. That epigenetic phenotype changes are maintained on cell-free DNA. And finally, that cell-free DNA epigenetic approaches can offer us better ways of phenotyping rejection more than the first-generation test, and they are potentially broadly applicable in conditions beyond transplantation. I'll stop there. Thank you.
I would like to thank all the speakers for an excellent and very provocative talks. And we have a few minutes for discussion. I would like to apologize if we cannot get to all the questions. We have excellent questions in the chat, but I think we can then post that responses in the emails or open discussion. So one of the questions towards Dr. Halloran was if in patients with DSA and mixed rejection, can the molecular microscope help to detect the AMR component?
Yes, it does. What we do is we separately analyze the ABMR-like component and the TCMR-like component, and then we score the tissue for the extent of each. So, well, when any process is mixed, it could be it's the old ticks and fleas problem. You could be you have mixed if you've got 1 tick and 99 fleas, or 99 ticks and 1 flea. So what you want to do for treatment is establish the components individually, and that's what we do, and we can do that. The significance of the ABMR component will be long-term. The significance of the TCMR component, especially in mixed rejection, it's really a devastating disease because the cardiac myocyte, the renal tubule, and the pulmonary tissue they remember having been attacked by a T-cell mediated inflammatory process. And I think we need more information about how to manage these really severe conditions. So the answer is yes, we can establish the components of each.
Thank you, Dr. Halloran. If I may ask a second question, and then I'll give it to John, and that would be to Dr. Brendan. I think I like us two questions, one from the chat and one from me. You showed us HLA and beyond. It was an amazing presentation of what can be done. The low-hanging fruit, I think, as pharmacogenomics, can that be applied in routine for managing immunosuppression? And the second question is regarding the target peptides defined with peptide array. Are they polymorphic in the allo or only just for self-peptides? Thank you.
Okay, so for the pharmacogenetics, there's a few layers I think to unpack there. So we are actually doing a project with Illumina where we're doing clinical sequencing, whole genome sequencing on pediatric transplant recipients. And in that setting, most of these kids are polypharmic. So, you know, they're on at least 5 medications. So even beyond tacrolimus, you know, we have a pretty good idea what gene variants would impact efficacy or adverse events. But if we take into account drug-drug interactions, there's we think there's a lot of value in trying to tie in the clinical covariates as well. So drug-drug actually being a clinical covariate. So there's a few layers there with the genomics to unpack. I think for tacrolimus guided dosing, it's still a little bit unclear in African Americans, especially with the additional variants around CYP3A5 and CYP3A4. But I definitely think there's going to be you know, the value you get out of a whole genome, not just for HLA or non-HLA, you will be able to tie it to genetics as well. And then for the peptide array, the beauty about printing these peptides, and I didn't really have enough time to kind of get into the weeds, but essentially we print 16mers, so 16 amino acids, and you can do any type of amino acid. You can even do modifications of the amino acid. There's actually about 200 amino acid when you include all the modifications. So you can put a polymorphism, say on position 4 and also on position 11. So you can basically tile at the depth you would like. So you could do 1 amino acid spacing across a given region of interest that you think may be allogenic or xenogenic. And then you could put in different polymorphisms as well. It's not a perfect technique by any means, but the good thing is you can look at scale at thousands and hundreds of thousands of markers.
Thank you.
Thanks. A question for Dr. Marvin. So you described single cell and spatial transcriptomics technologies. Do you envision these as being applicable to clinical diagnosis in real time, or are these things that would be integrated with bulk RNA sequencing or molecular microscope type technologies and help facilitate convoluting those signals to improve those diagnostic classifiers?
Yeah, I think that's a really good question. I thought about it a bunch after you assigned me that title. But I think that right now, I don't really see how single cell transcriptomics would be used clinically. You know, I do think it's too expensive and complicated. So right now, I think it's more of a discovery method, really uncovering biology, helping us understand things that we would otherwise not be able to glean from current data. But 2 things. So right now, I think it's more that information would get translated into a higher throughput technologies, such as bulk transcriptomics or flow cytometric sort of analyses of single cells based on the information we get from the single cell RNA-seq data. That said, you know, going forward, all these things are getting cheaper and people are getting better at doing them in a higher throughput way. So it's possible that we'll be able to apply these in a more useful way clinically.
And then, Dr. Akbarino, one last question for you that really speaks to how close are we in terms of getting cell-free DNA into clinical practice, and what are the ways in which it could be integrated with AMR diagnostics versus to what extent do we need to be improving the technology?
Yeah. So the first generation of cell-free DNA tests, which is based on SNPs, those assets are already commercially available. And there are companies that are providing them. I think working now with the FDA and CMS to make sure that those tests would be available to patients. That's number one. Number two, the newer generation cell-free DNA tests, I think they are still in development. They perform somewhat quite well. But I think we need to somehow figure out their finesse a little bit to improve the sensitivity of the assay. Those, I think, are a little bit far away from the clinic per se. But I think on the research front, seeing it in transplantation, maybe within the next year or so, it should be technologies that multiple groups can do in able to show that how it uses in transplantation. So I think the door is open. The door is opening for several of these technologies to be combined and used in transplantation. But this second generation cell-free DNA, I think we need to figure out some of the finesse and improve the sensitivity of the test.
Well, we're right at the hour, so I just wanted to thank everyone for joining this webinar sponsored by the ISHLTv Research and Immunology Professional Community. This session was recorded, and so the recording of the session should be online early next week. And also, everybody should be on the lookout for a survey about this session. It'll be coming by email. We'd really love to hear your opinions about this session and suggestions for future things that the research and immunology professional community can provide. Thank you.
Thank you all.